So how do electrical charges, these action potentials that are moving along your neuron, how do they actually get to the next neuron, to the next cell, or to the next cell, whatever the type of cell is, whether it's a muscle cell or a neuron? And the point at which the axon terminals relay the message, the electrical signal to the next cell is called the synapse, okay? So synapse basically means a junction with another cell, okay? Now, what we are, we have the, okay, we have basically we always have a case where we have this presynaptic neuron, okay? So presynaptic means before the synapse happens. So the neuron before you actually have the synapse. And this is your signaling cell, okay? You have your postsynaptic cell, which can be a neuron, or it could be a gland or a muscle. This is your target cell, and this is where the information will be transferred to, okay? We are going to talk about neuron to neuron synapses and neuron to muscle synapses. We will talk more about glands, I think, later on when we look at the endocrine system, okay? So for the rest of this section, we focus on neuron to neuron synapses and neuron to muscle synapses, which are known as neuromuscular synapses. Before we look at neuron to neuron synapses, let's just talk about the two major types of synapses that you get in animal bodies. And these are electrical synapses, where you basically just have a straight flow of electricity of your charge from one cell to the next. No conversion of the electrical signal. It just passes from one cell to the next. And it happens via what's called gap junctions, okay? So gap junctions, we haven't covered yet in this course. And if we had time, we would go into detailed cellular physiology, but we won't be able to. But basically a gap junction is a tiny little gap between two cells that allows only the smallest ionic particles through and does not let larger protein molecules in through the gap junction. Okay, so yeah, the electrical synapses, they're just a straight flow of electricity through these gap junctions into the next cell. And this is a very high speed form of transferring information, okay, from cell to cell. It turns out to be less common in animals for reasons we'll go into, but does have the advantage of being very fast, okay? Then we have chemical synapses, which, so chemical synapses are where you have an electrical signal coming from your neuron, and that gets converted into a chemical signal in order to get across this gap known as the synaptic cleft, okay? It's a little gap between your neuron and your next cell, okay? Your postsynaptic cell. And chemical synapses work via the release of what's called neurotransmitters, okay? Which we're going to go into in some detail. But for now, just to point out that chemical synapses are probably the most common form of, they are the most common form of synapse in the body, in animal, or especially in vertebrates. So they are slower than electrical synapses, okay? So this is known as synaptic delay. This is because the electrical signal needs to be converted into a chemical signal, which needs to then do its thing with the postsynaptic cell, which we'll show you the details of. And this all takes time, right? By time here, I mean like a matter of 1 or 2 or 3 or 4 or maybe 5 milliseconds. So our concept of time here has to speed up. When we talk about slow here, we're talking about like more than 1 millisecond. So although they're slower, they are very, they have the advantage over electrical synapses in that they operate in one direction only. Electrical synapses technically are at risk of running in reverse, okay? Running backwards, just because electricity can flow both ways, okay? If your charge differences are enabling that. So just understand that chemical synapses are a bit slower, they operate in one direction, and a major advantage of chemical synapses is they allow for various kinds of signaling events. So not just the excitation of a cell, a receiving cell, but also, for instance, the inhibition of a signal in a receiving cell. So we're going to come back to that, okay? So don't worry too much about it now, but understand that these are the advantages of chemical synapses. Even though they are slower, they have advantages. So this is just a visual depiction of a of where information, of where a graded potential signal, sorry, what I wanted to actually show you here, I'm getting a bit tired now, but we have these synaptic inputs, okay? So these are your, remember on your presynaptic axon, your neuron that's coming into your into the next cell, you have these axon terminals, and they, as I said, they almost look like they have little suckers on them, and they connect with the cell body, okay, of the next neuron. So now we're looking at neuron to neuron synapses, okay. These little synaptic axon terminal, they synapse with the cell body, and as you can see, there can be lots and lots of them. In fact, apparently there can be like thousands of them, okay? And they synapse with this cell body of this postsynaptic neuron, and they would pass information into it, which would be chemical transfer, and that would be converted back into an electrical signal that would carry on its journey along the axon of the receiving cell. Okay, I might not have explained this all very well, but just understand that this is what the situation looks like, and that we have our neurons attaching, basically synapsing with the cell body of the next neuron that they're about to stimulate. Okay, and then this is just a scanning electron micrograph showing what it actually looks like in real life. Always nice to look at things in real life. So here you can see that you have all your little axon terminals synapsing with a nice big sort of club-shaped cell body of the next receiving neuron, okay? So all these little sucker tentacle type thing are synapsing, okay? So for once, it actually looks vaguely similar to what it looks like in the diagram. Okay, just looking at neuron to neuron synapses, we are talking now about chemical synapses, okay? Chemical synaptic transmission. So let's just look to finish off, well, not finish the lecture, but almost finish it. We look at how a chemical synapse works, okay, in a neuron to neuron synapse. So we have what's called neurotransmitter, which is a a chemical substance, and it's stored in these little things called, these little bubbles that are called synaptic vesicles inside the synaptic knob of a presynaptic neuron. Now, this all sounds like a different language, but the synaptic knob you'll see when I show you the diagram, okay? Now, when you have an action potential reaching the synaptic knob, it basically causes a bunch of voltage-gated voltage-gated calcium channels, okay, to open, and this causes calcium to rush into the synaptic knob, which in turn, the calcium stimulates the release of these little vesicles, okay, these little bubbles that contain the neurotransmitter via a process called exocytosis. So it's all a bit difficult to follow when you have just text, but I'll explain this with a diagram. So chemical, just to mention that these kinds of chemical synapses are known as fast synapses, okay? They are rapid chemical synapses. This whole process is very fast, like less than 5 milliseconds for the signal to cross to the next cell. We will talk later in neuromuscular, with neuromuscular synapses, we look at slow synapses, which is a different kind of process. Okay, so just to say that when, as I was explaining, when the calcium ions rush into the cell and you get neurotransmitter diffusing out of the synaptic synaptic knob, out of the cell, into this gap called the cleft, you'll find that this then combines with the receptors in the next cell, okay, on the awaiting cell known as a subsynaptic membrane. And this binding triggers the opening of chemically gated ion channels in the subsynaptic membrane, and we will we'll give more detail about what that means in the next cell. But just to first look at, much easier to explain with a diagram, I think. But basically here we have our axon, of our presynaptic neuron. In other words, this is the neuron that is coming in and synapsing with the next, with the cell body of the next neuron, okay, down here. So this, right at the end, we have that thing that I said looks like a little sucker. That is the synaptic knob, okay? It's the axon terminal, okay? Now your action potential comes in here, it flows down your axon, it reaches the synaptic knob, and that's phase one, okay? Now phase two, calcium, the voltage-gated calcium channels are opened by this action potential triggers their opening, okay? And calcium ions rush into the synaptic knob, and step three, really good connection. They get on really well with these synaptic vesicles, these little bubbles that are filled with neurotransmitter chemicals. And because the calcium, because the calcium ions get on so well with these synaptic vesicles, they get all excited and they say, "That's it. Let's leave. Let's leave this place." And they want to get out of the synaptic knob now, and they leave. They leave the synaptic knob through exocytosis, which is a fancy word for the way these vesicles exit the neuron. And they, once they leave, they just release all this chemical substance, little chemical transmitters known as neurotransmitter molecules. Yeah, so at step 5, these neurotransmitter molecules fuse or combine with the receptors on the excuse me, they they combine with the receptors on these picture. These neurotransmitter molecules now, they've left this whole the presynaptic neuron, and they now combine or fuse with receptors. Okay, so for instance, here you see a little receptor. It's a little opening, nice little comfy spot for this neurotransmitter molecule to fuse with, to combine with, and once they fuse, this will cause an opening of the voltage-gated ion channel. And basically, we will follow this up by explaining what this causes in the postsynaptic neuron. But basically, in this process, I just wanted to explain that an action potential arrives from the neuron. It arrives at the end of the neuron, where it stimulates an influx of calcium ions, which in turn stimulate the release of the neurotransmitter molecules, which quickly travel across the synaptic cleft, this gap, okay, and fuse with these protein receptors that are sitting in the cell membrane of the receiving neuron, okay, and they then open the channel gates, and basically ion flow can now be altered in the receiving neuron. Okay, another animation which you can get in your own time. So you get two major types of neuron-to-neuron synapses. You get your excitatory synapses, okay, which are this is when basically the neurotransmitter combines with the receiving proteins, okay, in the next neuron, the postsynaptic neuron, and binding of this neurotransmitter opens what's called non-specific cation channels. Okay, so these are channels that allow the flow of these cations, these positive ions, particularly sodium and potassium. So that's what an excitatory synapse basically does. It opens up ion channels in the receiving neuron. Okay, so electrochemical gradients in these ion channels. Once the ion channels are open, electrochemical gradients favor the inward movement of sodium. Okay, this is just because sodium moves from a higher concentration and also electrically are attracted from the more positive exterior to the more negative interior of the receiving neuron. Okay, basically causes what we call a small depolarization of the receiving neuron. Okay, so by small depolarization, I mean that there's not enough of a reversal of the polarity to immediately cause an action potential, but it is nonetheless a lessening of the polarity. In other words, the charge becomes slightly less negative. Okay, so the excitatory synapse basically results in your receiving neuron, where these neurotransmitters have now moved to your receiving neuron, and they've bound with these channels, and they've opened them up, and they've caused a small depolarization. Essentially, what this causes is a graded potential. Okay, so this is where your graded potential that we'll come back to the beginning of the lecture, the graded potential that enters the dendrites and the cell body of a neuron. The graded potential is caused by this process, right? The small depolarization. Yeah, so the fancy name for this is an excitatory postsynaptic potential, or EPSP, okay? We will return to this term, so bear in mind EPSP, excitatory postsynaptic potential. Basically, a synapse that results in excitation of the receiving cell, okay? Inhibitory synapses are the next type of synapse we talk about. And this is where neurotransmitter molecules bind with your receptors sitting in the membrane of the receiving neuron, and they open what's called specific potassium or chloride channels, okay? So these specific channels they allow, obviously, the movement, the passive diffusion of either either potassium or chloride ions, depending on which particular channel is involved. And this okay, so for instance, if there's a binding with the potassium channel, this will cause an outward movement of potassium out of the cell. And if they bind with the chloride channel, then this will cause a movement of chloride into the cell, okay? Basically, what you need to understand here is that these inhibitory synapses result in a small hyperpolarization, okay? So instead of a small depolarization, we have a small hyperpolarization. In other words, the polarity increases to a more negative state in the receiving neuron, okay? And these are known as IPSPs, inhibitory postsynaptic potential. And they're very important in animal bodies, okay? Because sometimes you actually don't want to stimulate a neuron, you want to inhibit it, okay? You want to make it less likely that another action potential will happen in the receiving neuron, okay? And this is what an inhibitory postsynaptic potential produces, okay? It produces a state of hyperpolarization where the charge is more negative than the normal resting potential. So it might be like minus 80 instead of minus 70. And it just means that it makes it a bit less likely now that you're going to get further stimulation of that neuron happening. So just to point out that each of these sort of neurotransmitter receptor combination that I've been explaining, they always produce the same response, okay? So for instance, there's a neurotransmitter called GABA, which is short for probably some horrible long name, but GABA, G-A-B-A, is an inhibitory neurotransmitter in the brain, and there's lots of it going on in our brains apparently, and it's important in inhibiting a lot of stimulation that's going on. So sometimes you need to inhibit electrical flow, okay? So just carrying on with neuron-to-neuron synapses, it is very important to point out that after the chemical synapse has taken place and all of the stuff we've explained has happened, we need to remove the neurotransmitters from where they're sitting, hanging out in the synaptic cleft, okay? If we were to leave them there, the ions would keep rushing into the neuron, okay? And we can't have this continually happening. So it's important that very quickly, as soon as the signal's been moved onto the next cell, the neurotransmitters need to be removed from this synaptic gap, okay? And this happens by three major three major ways that this can happen. Either diffusion, so this is just diffusion along a concentration gradient away from the synaptic cleft. Then you get the neurotransmitters could be inactivated by a specific enzyme. In other words, they could be chemically altered so that they don't work anymore and they can't cause any further issues or problems, or they could be transported back into the synaptic knob, into the axon terminal, by what's known as reuptake carriers. Okay, so little bubbles, little vesicles that actually carry these neurotransmitter substances back to where they started. Okay, so back into the end of the axon. That's just the three major ways that we can remove these neurotransmitters from the synaptic gap. We will come back to this when we look, as far as I know, I think we will come back to looking more at how neurotransmitters are removed. I think when we look at neuromuscular junctions, neuromuscular synapses. Okay, so that is definitely more than enough for a lecture for this evening. It's like pretty late now. So, yeah, quite a lot to take in. So please read through your stuff, take notes, make sure you understand concepts more than small little details, rather understand processes. You know, how does a process work? How does a how does a chemical synapse work? In other words, how does electricity get converted into a chemical signal, which then stimulates the receiving neuron? How does this understand the process? You know, the action potentials arriving and the calcium rushing in, and then the neurotransmitters being released, and then the neurotransmitters fusing or combining with the receiving protein, which then open up channels for ion flow, which then affects the postsynaptic neuron. Okay, so it's like a whole process, things of events, sequence of events, things following each other. And if you understand these processes, you'll find you'll do well in understanding most of the stuff in this course. So focus on processes, understanding the sequence of a process, and the concepts involved. Okay. Yeah, just to finish off with a quick video, just showing quickly a 3-minute video that shows roughly how synapses work. Okay, so thank you for joining, and I'm going to show the video, and then that will be the end of the lecture for today, and I'll post another lecture tomorrow. Okay, so just bear with me quickly while I try to get the video to work. So to do that, I need to quickly turn off the microphone.
Brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. In the sending neuron travels down the axon until it reaches a synaptic terminal. Between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, It causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.